Skin barrier structure and its functions. The skin barrier structure and function is essential to human health. Unrecognized functions of epidermal keratinocytes show that the skin plays an important role in adapting to the whole body and to changing environments. Skin microbiota plays an integral role in maturation and homeostatic regulation of the keratinocytes and host immune systems networks with systemic implications. The primary interface with external environment and the biodiversity of the skin habitats is heavily influenced by the biodiversity of the ecosystems in which we reside. The maturation and function of the systemic immune system in a young child is dependent on contact with microbes. This has implications for the development and function of virtually all organ systems, including the brain, which are all profoundly influenced by the immune system. Locally, microbial immune interactions in the skin are vital for optimal barrier function, pathogen defense, and tissue repair, with the production of key anti-inflammatory and antimicrobial compounds to maintain healthy tissue of homeostasis. Just as the gut, the metabolome in the skin reflects the combined functional metabolic activity of the microbes and our host tissues. While the effect of delivery method on an infant gut flora persists of over the first 3 to 6 months of life during the critical windows of the immune system development, delivery method has a major influence on initial colonizing bacteria in all habitats, including the infant skin. Unlike vaginal delivery, anally delivered infants who are colonized by the bacteria from the vaginal community, infants born by C-section are dominantly colonized by staphylococcus and other taxa-reflecting maternal skin flora. There are clear evidence that both innate and adaptive immune function in the skin are influenced by the commensal skin microbiota, due to the inhibition of the pathogens, inflammation, immune development, and homeostasis, repair and angiogenesis, and T-cell differentiation. Increased substance P is linked to eczema and barrier dysfunction. The skin manufactures and metabolizes steroid hormones, peptide neurohormones, and neurotransmitters, including some which are further disseminated by sweat and semen. These chemicals make contact with cutaneous microbes and influence adhesion, growth, and virulence. For example, experimental studies provide a pathway between psychological and stress-induced increases in local substances, changes the skin and microbiota. Recent twin studies indicate that microbiome is a product of both genetics and shared environment. Strong associations have been found between the composition of skin microbiota and genetic factors related to the skin barrier and its function. As with other organs, the skin innate immune system is a composite unit of interacting human and microbial elements. An establishment of commensal microbiota is in developing initial homeostatic control of skin immunity. Skin stressors include dryness and barrier disruption and have been shown to stimulate cutaneous cortisol production, and this action may be mediated through activation of inflammatory cytokines. New research places epidermal keratinocytes at the forefront of sensory systems, recognizing that they generate a variety of hormones and neurotransmitters that influence whole body states and even emotions. This includes not only the senses of mechanical stress, temperature, and chemical stimuli, but the capacity of glucocorticoid production. The signatures vary with changes in local microenvironment between individuals with health, behaviors, and environmental contexts. Cytokine production. Many of these actions are mediated through the activation of innate immune receptors or keratinocytes and other local immune cells. As two different strains of the same bacterial species can have profound functional differences, there is a critical need to advance research in this more functional direction. Although less is known about other resident microorganisms such as viruses, fungi, and parasites, they are likely to interact with the wider ecosystem and influence cutaneous immunity. There are 4 phyla bacteria residing on the skin, and they mainly fall into Firmicutes, bacteroidetes, Protobacteria, and Actinobacteria. Within the bacterial groups, strain level identification remains obscure. Once initiated, the chronic inflammation can itself promise the normal production of SC lipids, which means that the cutaneous microbes may sit squarely within any discussions of the human epidermal barrier. There is much focus in a good microbiota in this context. We argue that the role of skin ecosystems may be equally important, especially as it affects in the skin integrity, such as eczema, has increased. The importance of very early life events in establishment of the skin ecology and homeostasis in the pathways to disease. It provides reason for optimism in the search for prevention and personalized medicine and effects for strategies for the treatment. Inflammatory diseases is informative because allergy is one of the earliest manifestations of inflammation, often first observed in the skin as disruptions in the barrier and function in atopic eczema. The declining microbial diversity that has been long linked to the rise in an allergic disease also has important implications for other organ systems across the life course. These studies have identified three broad microenvironment types with characteristics of the microbial communities: the sebaceous areas, the moist areas, and the dry areas. The sebaceous areas is where the Propionibacteria species and Staphylococci species predominate. The moist areas where the Corynebacteria species predominate, with Staphylococci also present. The dry areas with mixed population and greater prevalence of B. Proteobacteria and Flavobacterial. These unrecognized functions suggest that the skin plays an important role in adapting whole body physiology to changing environments and raises new questions about the impact of the local skin microbiome. While the skin is arguably one complex ecosystem, it is made up of many different habitats and microbial communities. In any individual, the skin microbial composition is highly heterogeneous and depends on the local microenvironment of the specific skin site. Everything that people touch, bathe in, and breathe and eat and drink is reflected in their many microbial ecosystems, including the skin. This underscores the important influence of environmental conditions on colonizing microbiota at every age, but particularly early in life, when various surface ecosystems and immune pathways are being established. It also provides another mechanism by which nature relatedness, biodiversity of ecosystems in which humans reside or to which they are exposed. Rural environments can have beneficial effects on physical and mental well-being. In other words, a person's level of connectedness with the natural world. With that being said, that is the skin microbiome and the systemic immune regulations. I hope you enjoyed this video and stay tuned to what's next.